All right.
I know, I was like, what I'm gonna do, like, this Shamilina? I was like, dude, can you
All right, let's go ahead and get started. I'll just sit and talk to myself. I don't have anything really to announce this morning, or this afternoon. See, told you I'm tired. Friday's assignment is a group wiki, just so you know ahead of time. Okay. We're gonna move into chemical control of growth on Friday. So each member has to. There's like 8 different terms or 8 different categories of chemicals. Each person has to do 2 of them. Give an example, and I can't remember what the rest of it is. But follow along with that, and yeah, that's all I need to say there. Other announcements coming on Friday about stuff going on next week. So, I wanted to start off where we left off on Monday with this problem. So, just given the prompt, you have isolated a microorganism from the hull of an ocean liner. What has to be true about this organism's growth?
It likes salt.
It likes salt, so you have to have salt in the media. Therefore, it is what? What's the term that we would use if it likes salt? Halophile, or you could say halotolerant, given at least what we know right now, right? If we add salt to the media, does that make the media selective or differential? It is selective at this point. And again, what we mean by that term is that selective media will select for a certain type. So oftentimes, selective media, we're talking about selecting for one of these growth characteristics. Growing cells at 30 degrees Celsius is in some ways a selective condition. It selects for those mesophiles, to some extent psychotrophs, but not allowing for the growth of hyperthermophiles, thermophiles, or psychrophiles. Okay. So this one would be a selective media. What would it select against? What wouldn't be able to grow in media containing 3% salt, which is roughly the percentage of salt in salt water, ocean water? What wouldn't be able to grow? Sounds like a future exam question. All right, I'll just move on. No, I'm not going to give the answer. You guys can tell me.
Wait, ask the question again.
In 3% salt, what is that media selecting against? What won't be able to grow in 3% salt? Sarah, that will be. A non. So in 3% salt, a halophile would be able to grow.
I thought they have to have a lot of salt.
Some can grow. So halophiles have a range. There are some like Vibrio vulnificus, which is a saltwater organism. Between like 1 and 5% it will grow. But there are others which are like extreme halophiles where they need like 10, 15% or higher. A halotolerant would grow as well. Usually they'll grow anywhere from like 0 out to 6 to 8% salt typically. But our non-halophiles are ones that in the absence of salt would be able to, it would only grow in the absence of salt or really low concentrations of it. All right, so let's go back to this problem real quick. All right. So I think in this one we said the concentration of this initial was 2.67 times 10 to the 4th CFU per mil, or 26,700. And again, I don't care which way you personally want to express that value, whatever works best for you. What did we get for the second one? 3.2 times 10 to the 7th, or 32 million if you're writing it out. How about for the last one? 4,100,000, or 4.1 times 10 to the 6th. Okay. The reason why I like the exponential is you can do a really quick check here, just looking at those exponents to figure out which one grew the best. You don't even need to look at the rest of this. If you just pay attention to these exponents, which one is the largest? Yeah, middle one, 10 to the 7th. So using this, then, how would we classify this one based on the temperature at which it grows best? So given those three temperatures, where it's got the most growth, how would we classify it? It's a psychro something. What's the difference between a psychrophile and a psychotroph? Psychrophiles love cold, and psychotrophs are like middle. So psychotrophs usually, they can grow at refrigerated temperatures, but they do so very slowly. They usually grow best at around room temperature, maybe a little bit above room temperature. So this one, based on its growth characteristics, we would probably classify as a psychotroph. Okay? How do you inhibit the growth of a psychotroph? Bingo. Burn it. Incinerate it, right? So in order to control microbial growth, what we're moving into today is we've spent time talking about how to measure growth. We've spent time talking about how do we classify growth or classify organisms based on their growth. Today we're moving into how do you stop that? Because especially for those going into the medical profession, this becomes a pretty important question. And oftentimes we're concerned with inhibiting the growth of mesophiles. But there are many ways in which we can inhibit microbial growth. And today we're just going to focus on those physical methods. So for psychotrophs, burn them, incinerate them, heat them up above 37 degrees Celsius. Okay? Here's what I want to do. All right. Who had desiccation as their particular definition to define? Raise up those hands high. Okay. You're going to meet in the back corner. You're going to talk about UV radiation. Okay, and I'll give you the instructions of what you have to do in a minute. Who had UV radiation? Okay, you get to talk about desiccation. Who had filtration? You get to talk about pasteurization. And who had pasteurization? You get to talk about filtration. Okay, so you're talking about something that you didn't do your particular one on. So what we're going to do, and we're going to do this in big groups. I'm switching it up from what it was this morning because it didn't go well this morning. I'm going to hope that this works a little bit better.
We different.
I know you are. I totally know you guys are different. For a couple of reasons, but we'll leave those reasons to be guessed upon right now. So, if you were discussing desiccation, back corner. If you're discussing filtration, that back corner. If you're discussing UV radiation, this front corner. Discussing.
Wait, which one is desiccation?
Back corner. And you're talking about pasteurization over here. What you need to do before you move, you need to, as a group, come up with a description of that, and then in like 2 or 3 minutes, come up with as many examples as you can. Go.
So if I had this desiccation on the back.
Did you have it to explain? So now you're talking about, I can't even remember. Radiation, so you're up over here. Yeah.
Let me go be with my people.
No. The definition of desiccation or a description of it.
Pulling water away from the organism, and that inhibits the growth of the microbacteria.
Perfect. And reason being, water is required for pretty much every metabolic activity. Okay, so that's the essential reason why. Examples, you guys focused on food ones primarily, so what are examples of it?
Like dried meat, like beef jerky, dried fruit.
And other dried things?
Drying.
Fish. if you were on a ship back in the whaling days, right? Okay. Ostrich jerky, if you've ever had dried ostrich jerky. Sun-dried tomatoes, etc. Okay. Other places where we might see this desiccation, but you may not even realize it. Jams, peanut butter, those are ones where the desiccation isn't really so much from like a drying out of something, but there's high enough salt or sugar concentrations to actually pull. It's a hypertonic solution. It pulls the water away from the microorganisms. So we can see that as an example of desiccation, or that high osmolarity as well. One of the other things to point out, and you guys don't answer this since we already talked about it, as something becomes desiccated, water gets lost, what happens to osmolarity or salt concentration? It increases. So as water concentration goes down, that naturally increases the salt concentration. So organisms that survive desiccation really well are typically ones that also can survive high concentrations of salt, for example. Your halotolerant, or particularly your halophilic organisms. Okay. Anything else that we're missing? No. Okay. Filtration. What's a description of filtration?
Filters out large particles or microorganisms from fluids.
Okay. What's the typical size of a bacterium?
Small.
Numbers, please.
0.05 micrometers.
That's a little small, but that's actually the size of a typical filter.
It's about one, isn't it?
Yeah, it's like a micrometer or so. So for filters to work to inhibit bacterial passage, usually those filters need to have a size restriction below the size of the microorganism. 0.2, 0.45, something like that. Or typical sizes, 0.05 even works too. Anybody use like a life straw or anything like that? How do life straws work?
Stick it in the water.
Do you know the what's actually happening there? Like what's inside of a life straw?
There's a filter within the life straw.
Yeah, I mean basically it's a bunch of like small tubing that has, and their rate for like the original life straw, the pore size is 0.2 or something like that. The goal is to remove, as they say, 99.9999% of bacteria. Okay. Yes. Yes. What are other examples of filtration?
Instagram.
What's that?
You asked.
I did, but any others?
Dialysis.
Yeah, actually, dialysis is a perfect example. That's a really good one. Nice job reading. What organisms aren't going to be inhibited by common filtration? Anything smaller than your pore size. Viruses would be one. Lipopolysaccharide, sometimes it can't be filtered out, and we'll talk about later about the toxigenic effects of lipopolysaccharide. Prions. So anybody a hunter in here? Of deer, particularly? Zach, what disease do you have to watch out with deer?
I can't think of
CWD.
CWD, yeah.
Chronic wasting disease. It's a prion disease. Basically, it's driven by proteins. Proteins are too small to be filtered out as well. Radiation.
UV radiation is a method that produces short wavelengths to kill like surface bacteria, and it destroys nucleic acids, which disrupts the DNA and doesn't allow it to.
Okay, good.
And then do you want ionizing?
Yeah, let's do ionizing too.
Ionizing radiation also damages the DNA and produces long wavelengths to like penetrate inside of like not just surface area, but like inside of.
Right. So the major difference between the two of them is really the ability to penetrate through a solid surface. So UV is really good for superficial, topical type treatment. Ionizing, penetrating radiation. Both are commonly used with water as well. So like Newer sewerage districts use UV radiation as a way to treat their water instead of something like chlorine or something like that. Anything that's going to survive that? I can think of 2 examples. What did we say is a type of structure that's incredibly resistant to a lot of different stresses?
Spores.
Spores, endospores. If they're not replicating, spores, because of the way that their DNA is bound up and because of that spore coat, are pretty resistant to UV light. Still affected, but less so than the vegetative cells. There's also an organism known as Deinococcus radiodurans, which is the most UV tolerant organism that we know. I don't understand the importance of it other than it's really cool to study. So,
Is it dangerous to humans?
what's that?
Is it dangerous to humans?
You tell me. Is UV light dangerous to humans?
No, I'm talking about that bacteria.
Oh, no. No. No.
So if you coated
it's-
yourself with it, could you be UV light resistant? Prevent
You know
sunburns,
what? There
prevent-
actually, over the summer, there was a group, I saw a report that they're using DNA as a new sunscreen. And that they think that it would be something that would last many, many years, actually, instead of having to reapply.
The
Anyway.
team's gonna be rich.
Yeah.
Got
Yeah.
it.
All right, last one. You guys had pasteurization.
The rapid heating and cooling to kill harmful bacteria without damaging the food.
Good. What's it work? What does it? What are examples of where we see it?
Like food examples are butter, cheeses, cream, vinegar, almonds,
Good. Good.
commercial sauerkraut.
Fair enough.
Are we making sauerkraut?
No. Well, we'll talk about that later down the road. Perfect. Perfect. And then what's going to be resistant to pasteurization? Hyperthermophiles, thermophiles, but those, at least to us, are not of medical importance, right? They are ones that, given the temperatures that we reach with pasteurization, typically it's not high enough to kill them. And one of the things about pasteurization is it's not designed to kill everything. It's designed particularly to kill pathogenic microorganisms. Is it 100% effective? It definitely does kill the pathogens, but there are some other ones that may survive that temperature burst. Okay, any questions on any of these 4? All right, go back to your seats. We're going to do some comparing between the different ones.
So then the UV is I got super
All right, so this here is an example of an autoclave. We have one of these upstairs in Sonderegger. Basically, all the media that you guys use, with the exception of a couple, go through this autoclave or one like this in order to sterilize. And sterilize means what? When we say something is sterilized, what do we mean by that? Kill everything. Exactly. Kill all living life altogether. Autoclaves get up to about 120 degrees Celsius on the inside. It uses steam heat as a way to kill. Wet heat works much more efficiently than does dry heat. Just in the same way that if you have 100 degrees Celsius in Orlando, it feels a heck of a lot more disruptive than 100 degrees Celsius in Arizona does, because of that humidity factor there. How does autoclaving differ from pasteurization? Sarah?
It doesn't. It's like not only like really high temperature, so it's really high pressure.
That's true, and one of the things we oftentimes forget about with the autoclaving is that some organisms are actually really sensitive to that pressure buildup as well. So that is definitely a difference between the two of them. What else? Time of exposure is much different. Shorter for pasteurization than it is for autoclaving. How does that affect then the end result?
In pasteurization, not everything is killed.
Yeah, pasteurization isn't necessarily a sterilization. Pasteurization is a disinfection, really. Its goal is to remove harmful pathogens, but it doesn't remove all pathogens. When we consider inhibiting microbial growth, a couple of things I want to point out real quick. One, growth doesn't necessarily, or inhibiting growth doesn't necessarily mean death. We can have conditions like pasteurization where by heating for a short period of time, it might kill the majority of microorganisms. It might stunt the growth of some others or damage them for a period of time in which they have to recover from that and then can begin to grow again. And oftentimes the inhibitory effect is dependent on a couple of different factors. One, the degree of the stress. So for example, for a for a non-halophile, they're going to survive and grow better in 2% salt than they will in 10% salt. So that greater concentration of salt will do more inhibitory. In UHT pasteurization, we still do find that there are microorganisms in there after that treatment. Whereas autoclaving, 120 degrees Celsius for 20 minutes, that kills everything, including spores. Okay. Other differences between these two different physical methods. Those are kind of the major ones. The method that we see most frequently right now is the 72 for 15 seconds. This is what you would find for typical milk. If you've ever had, for example, like nowadays they sell chocolate milk in like what would be equivalent to like a juice or a juice container. Some of those aluminum foil wrapped ones, they're at room temperature. Those ones are ultra high temp treated. So they have longer shelf lives and you can actually store them at room temperature, whereas just by typical methods we still refrigerate. There was one more thing I wanted to point out and I can't remember what it is. Damn. Okay. How about autoclaving versus filtration? Similarities, differences. Go ahead, sir. No? Okay. Yeah, so, and the term that we use, I would never fault you for saying killed with viruses. I consider them living, but some people don't. The term that often gets used is inactivated. So that basically that heat denatures the proteins of the virus and they're no longer able to infect a cell with autoclaving. With filtration, they aren't exactly inactivated. They might make it, if the pore size of the filter isn't large enough, they can get through that filter and still cause disease. What else? In the same vein, bacteria aren't really killed by filtration either, are they? What's different is that the filtration is a physical method of separation, absence of heat. What's the benefit of filtration then? Why do we use it? Relatively, yes.
It's not really changing the chemical makeup.
That does have some importance to it. So it doesn't necessarily change the chemical so much. Sometimes we can find that heat, for example, with antibiotics, heat's a bad thing. If you look at any bottle of medicine that you may have at home, it says to keep at like between room temperature, don't let it go above 90 degrees Fahrenheit. They prescribe that because above that temperature, the chemical nature begins to change. you might lose some of the chemical composition that makes up that that drug, for example. So antibiotics are, liquid suspensions of antibiotics are filtered. Pretty much anything that is probably UV or delivered by IV would be filtered rather than, or treated with something like UV light rather than autoclaving it. Some solids cannot be autoclaved. Obviously, you cannot autoclave a table; it is too darn big to even fit in there. Plastics, some plastics can't be autoclaved either, and so we have to have other methods to treat those. UV light, some gases are used sometimes in chemical regulation of these treatments. All right. Sarah.
Why is there a cooling portion?
Say that again?
Why is there a cooling?
That's a great question. So the reason why there's a cooling step here, or a really quick snap cooling, is because imagine if you heat this and then allow it to just like collect in some container and cool over time. What's going to happen is that as that temperature starts to drop, it's going to pass through a range that promotes mesophilic growth again. So in order to get around that as quick as possible, or in order to prevent microbial growth from occurring, that snap freezing or that snap chilling would prevent that growth of mesophiles from ever occurring. Does that make some sense? So just as a way to get them as cold as possible, as soon as possible, so that those mesophiles and psychrotrophs don't end up actually replicating during that chilling process. Why does milk still spoil? There's still living stuff in there. What kind of organisms are likely to be the ones that are spoiling it? If we were to define a characteristic of them. So what kind of organisms grow at cold temperatures? Psychrophiles, possibly. What else? Psychrotrophs, likely to be the ones that contribute to that spoilage. Good. All right. Covered that. We've talked about... Oh, the last thing I want to mention here, of these different methodologies, most of them affect metabolic activity. Chilling slows down metabolism, slows down enzyme rates. Heat, desiccation, both will affect protein structure and lead to denaturing. And UV, on the other hand, is going to lead to DNA damage, as will ionizing radiation, just to kind of give you an idea of what's affected here. All right, let's change focus. How would you, I'm going to give you two different microorganisms. How would you inhibit the growth of a mesophile in a chicken thigh? Whoop. I can't spell chicken, apparently. And then the second one is, how would you inhibit the growth of an acidotolerant halotolerant thermophile? So 2 minutes, quick thought. How would you inhibit the growth of these two different types of microbes? Go.